Good evening, and welcome back to Vibing Science. Tonight, we journey into the heart of life itself, not by meticulously reading every single word of its intricate instruction manual, but by observing its unique fingerprint, its silent code, through a revolutionary lens. Prepare to immerse yourselves in a tale of ingenuity, where simplicity meets profound insight, and where the very essence of an organism can be unveiled without ever uttering a single genetic letter. For generations, the dream of understanding life's blueprint, the genome, has captivated scientists. We've sought to categorize, to identify, to unravel the evolutionary threads that connect every living thing on this planet. And for a long time, the only path we knew was through sequencing, a painstaking, often slow, and resource-intensive process of decoding the precise order of adenine, guanine, cytosine, and thymine. But what if there was another way? A faster, more elegant method to grasp the fundamental differences and similarities between organisms, without the need for that exhaustive, letter-by-letter translation? Imagine standing before a vast library of ancient texts, each one representing the complete genetic instruction set of a different species. For centuries, our approach has been to painstakingly transcribe every single character of every single book to understand its contents. But what if instead we could simply look at the unique binding patterns of the ink on the parchment, the specific way the paper folds, the very texture of the material, and from these subtle cues discern not only which book belongs to which author, but also how closely related one text is to another? This is the essence of what we call genome profiling. Pioneered in the early 1990s by the brilliant Japanese biophysicist Professor Koichi Nishigaki and his dedicated colleagues at Saitama University, genome profiling, or GP, emerged as a paradigm shift. Initially, it was called DNA profiling, but to avoid confusion with a rapidly developing forensic technique that also used that very same name, it was wisely rebranded. And with this new nomenclature, a new era in biotech began to quietly unfold. At its core, genome profiling is a masterclass in elegant simplicity. It doesn't ask us to read the entire story, but rather to observe the unique way the story's pages melt and reform under specific conditions. Think of it like this. Every double-stranded DNA molecule, with its intricate helix, has a characteristic melting point. As you gently increase the temperature,